Welcome everyone to the REC session. And as Hannah mentioned, we're going to have a session this afternoon as well as tomorrow morning. And what I'm going to do here briefly is just introduce our committee as well as, sorry, I think I went off video here. Yeah, I'm going to introduce our committee and tell you a little bit about our initial goals. This is the inaugural steering committee. And then you're going to hear from junior faculty and trainees from individual ADRCs. And as you'll remember, perhaps from prior years, this is just expected to be a wonderful session. It's very inspiring and enlightening to hear what's going on at the individual Alzheimer's disease research centers. So I am the REC leader at the University of Wisconsin, and I'm chairing this year. Our other members are Dr. Hussein Yassin, who's at the University of Southern California, who will be chairing in year 2, as well as Dr. Elizabeth Head at the University of California, Irvine, and she'll be chairing in year 3. And I'd also like to acknowledge Cheryl Spenceley, who's the REC coordinator in Wisconsin and has really just been such a great help to our committee. So we identified 3 goals in the first couple of meetings that we had as a steering committee. Our goal 1 is to increase collaboration across the Alzheimer's centers and other organizations to support networking and exposure for trainees, learners, and scholars at the individual Alzheimer's disease research centers. And one of our main initiatives here is to develop a website, and I'll tell you on the next slide sort of how we're going about that. But ideally, the goal is to share resources across the ADRCs as well as increase networking opportunities. Our goal 2 for the RECs is to develop a REC collaborative core curriculum. The idea here is not to mandate a curriculum that's going to be uniform across all of the centers. We realize that all of the RECs are engaged in different and wonderful activities. The idea here is more to just leverage all of the expertise that's available at all the Alzheimer's disease research centers and make training materials available across the centers and really just facilitate the sharing of these materials. And we anticipate that the website could be a potential way to do that. And finally, goal 3 is to promote diversity in ADRD research. So that's really going to be at the forefront in everything that we do. So you heard from Amy Young a little bit earlier that we are working with NACC, and I just want to thank the staff and leadership at NACC for working with us. The idea is to create a hub on the NACC website that's going to house REC trainee resources to support career development of our trainees and scholars. And just a few brief points. This is going to be hosted on the NACC domain. Our steering committee will be involved in helping to launch this website, but it'll be upon future steering committees to help maintain this. We want it to be accessible, low maintenance. We would like to start out simple, which means that it's really kind of goal number 1 is to explain what the RECs are. Who we are, what we do, what the mission is. We anticipate having REC leaders and their bios featured on the website, also highlight trainees and have a job board, for example, so that we can help people find new individuals when there's opportunities, as well as help new graduates find jobs. So we think this will be a really useful resource. We think there's a lot of things that could be done via the website. And so actually NACC will be reaching out to representative learners from each ADRC to do a needs assessment, to do kind of a focus group and learn from individuals who would benefit from this website, what kinds of things that they would like to see there. And ultimately, we would also like to use this to house materials from ADRCs and to, as I mentioned, increase the accessibility of materials and curriculum materials. We would like to hear from all the REC leaders at the other ADRCs, and I just want to say thank you to everybody who's reached out so far with ideas and also offers to help. There's a REC leaders meeting that's planned for the summer. The date is to be determined, but it will likely be in July or August, and we'll make sure to not overlap with large upcoming meetings. But the idea will be to gather your input and feedback about what the priorities should be for the RECs. We're going to disseminate a survey, so that will be emailed to the REC leaders, but feel free to share it with others at your site who are involved in the RECs and provide your feedback. Yes, and I think that's it for me. I'm going to now pass it over to Dr. Yassin.
Thank you, Barb, for this nice introduction, and welcome everybody to this meeting. We're so excited that we have a group of outstanding scholars presenting their work in front of the National ADRC Spring Meeting. You know, we've had a difficult time choosing the best presentations. We truly enjoyed hearing and seeing the work that you have been doing all across the country. We have decided to score the best presentations that we thought would be of interest to the general audience and broke them down into 2 days. I will be making the introduction today, and my colleague, Dr. Head, will be making the introduction tomorrow. Now, bear in mind that we would like you to score these presentations and, in a sense, choose your top 2 choices. There will be an opportunity there's a link that's going to be shown in the next slide where you could select the top 2 choices from today and the top 2 choices from tomorrow. And tomorrow at 1:45 p.m. Eastern Time, Dr. Andy is going to announce the winner of the REC Scholar presentations. So can you move to the next slide, Barb? Do you have the option? Yeah, so we've got you know, a large group of presenters. Some of these presenters are presenting today and tomorrow, but there are others which are not presenting, and there was a link provided to watch all these presentations in the chat box if you would like to see that. We acknowledge all these presentations. Next slide, Barb. So today, Jeiran Choupan from USC is going to start the presentations, followed by Kimberly Alonge from University of Washington, Renaud Lajoie from UCSF, Shirley Wu from NYU, followed by Bryce Mander, Valentina Ghisays, Daniel Ohm, Robert Turner, and Karina Alvinia. I hope I did not butcher your name, so I apologize if I did not pronounce them. But we're very excited to see all of these presentations. And again, please remember to vote. The link is present on the site and in the chat box after seeing those presentations, and help us select the top presenter for an award that REC will be announcing tomorrow after the directors meeting.
Okay, with that, Barb, did you have any more content, or were you ready to start the presentations?
Yes, and Hannah, I think you're starting them from your side.
Yep, I will go ahead and queue up the video now.
My name is Jeiran Choupan, and I will be talking today about some of our recent work at University of Southern California on mapping brain clearance system in early cognitive decline and Alzheimer's disease. Glial lymphatic system is a system that is involved in the clearance of metabolic waste, which consists of perivascular tunnels, allowing glial lymphatic transportation, and is important for brain homeostasis. It has been shown that the alteration or dysfunction of this clearance system could contribute to Alzheimer's pathology. In a schematic view, when there is dysfunction of the clearance system, all metabolic waste, including amyloid, may aggregate within the perivascular space, contributing to Alzheimer's pathology. The clinical manifestation of dysfunction of the clearance system is the enlargement of the perivascular space, or PVS for short. Therefore, we hypothesized to observe higher PVS presence in MR image in mild cognitive impairment compared to healthy control. And we examined if PVS alteration is non-uniformly distributed across the brain. We started by identifying the current clinical routine. The conventional approach is manual categorical scoring system in different parts of the brain, like central semioval of white matter. To this end, a neuroradiologist will look at an axial slice of the brain, for example, around central semioval. They may adjust the contrast a little for better visualization of the PVS closely, and then they start counting the PVS. But the problem with this technique is that it is very laborious and non-specific. Therefore, we have developed an automatic technique by first improving PVS contrast by combining T1 weighted and T2 weighted, and then applied a specialized filter to segment the PVS. We then started looking at PVS in Alzheimer's disease and assessed our hypothesis. We looked at data from ADNI-3 and USC ADRC. We started focusing on early cognitive decline by comparing MCI versus healthy control. We used T1 weighted images to map PVS. In this case, we didn't have T2 weighted to enhance the PVS, so that was the limitation from the clinical data set. Since participants are older adults, detecting PVS from non-PVS hypointensities in T1 weighted, such as white matter hyperintensities, were difficult. Therefore, we used FLAIR to remove non-PVS lesions from the PVS maps. We also created normative template across cohorts. In this example, I also included the AD group. In this map, brighter color corresponds to higher percentage of PVS in white matter. We see a significant increase in AD versus controls. Interestingly, we also notice a lower amount of PVS in anterosuperior part of the medial temporal lobe in MCI and AD, which was in reverse direction of our hypothesis. This suggests that enlargement of the PVS is not the only pathologically relevant feature of the clearance system, and the presence of the PVS pathway across the brain could play a role in physiology and pathophysiology of the brain. We also noticed that for the difference in PVS that we detected in the anterosuperior part of the medial temporal lobe, the effect size is almost as large as the differences in the tau SUVR in entorhinal cortex, which reflects Braak stage 1, and larger than global amyloid SUVR differences between cognitively normal and MCI. Therefore, it is likely that what we are observing here is an early feature of the Alzheimer's disease. The observed PVS difference in anterosuperior part of the medial temporal lobe was significantly and negatively correlated with tau SUVR in entorhinal cortex. In addition, we found PVS differences was independent of amyloid and independent of APOE4. It was larger in female MCI compared to male, and also larger within individuals with history of hypertension. Currently, we are working on a natural history study that aims to map normative PVS across lifespan, funded by Brain Initiative. Thank you for your attention.
Hello, my name is Kimberly Alonge, and I am an acting instructor of medicine at the University of Washington, and I am representing our Alzheimer's Disease Research Center. Today, I will be sharing with you my research regarding changes in brain matrix extracellular matrix sulfation in Alzheimer's disease. Perineural nets are a very specific extracellular matrix that enmeshes and regulates circuitry involved in memory and cognition. Interestingly, phospho-tau accumulation occurs primarily in neurons devoid of these PNN matrices. Furthermore, patients with Alzheimer's disease exhibit a loss of these PNN structures evidenced by a decrease in their histological staining. Overall, this suggests that PNNs may be protective in the AD pathogenesis, and studies that investigate their loss are thus warranted. Function and stability of PNN matrices are dictated by the incorporation of 5 biologically relevant chondroitin sulfate isomers, including a non-sulfated O isomer, a monosulfated A and C isomers, and disulfated D and E isomers. As you can see, each isomer associates with a very specific biological function that then regulates and influences PNN function and stability. We hypothesized that changes in the brain matrix chondroitin sulfate sulfation code may contribute to PNN loss in Alzheimer's disease. And to test this hypothesis, we utilized state-of-the-art mass spectrometry analysis to analyze the relative abundance of each chondroitin sulfate isomer from postmortem brain tissue. Our postmortem cohort included 16 non-demented controls and 15 Alzheimer's disease patients that were sex, age, and PMI matched. We found that chondroitin sulfate isomers isolated from the middle frontal gyrus of AD patients exhibited significant alterations in their chondroitin sulfate sulfation code, including a decrease in the O isomer and increase in the PNN destabilizing C isomer and neuroinflammatory associated E isomer. When we investigated whether these isomers were changing in the cerebellar cortex, a brain region relatively spared in AD, we found no change in any of the isomers. This result suggests that changes in brain matrix chondroitin sulfate sulfation patterns occur in brain regions vulnerable to AD. To investigate the timing of these changes, we correlated the chondroitin sulfate isomer changes to the 3-category progression of REC staging. We were very surprised to find that changes in chondroitin sulfate isomers occurred both prior and after AD clinical pathology. Specifically, changes in the O isomer and increase in the PNN destabilizing C isomer and neuroinflammatory E isomer occur incrementally both in our non-demented patients as well as our AD demented patients prior and after phospho-tau accumulation. An association with phospho-tau confirms this result. Specifically, we found that phospho-tau negatively correlated with a decrease in the O isomer, but positively correlated with an increase in the PNN destabilizing C isomer and neuroinflammatory E isomer. However, when we associated these changes with cognitive function by associating isomers with the MMSE scores, we found that only the decrease in the non-sulfated O isomer and increase in the neuroinflammatory E isomer associated with a decline in cognitive function. And we believe this is primarily due to a hypersulfation of the chondroitin sulfate glycans. Overall, our model shows that changes in chondroitin sulfate sulfation patterns in PNNs are occurring both prior and after AD clinical pathology. Specifically, our non-demented population showed hypersulfation of PNN CS GAGs, and our AD demented population showed significant increases in the PNN destabilizing C isomer and neuroinflammatory E isomer. I'd like to thank University of Washington, the ADRC, and my sponsors. Thank you very much.
Hi, my name is Rajwa. I'll be presenting about the heterogeneity of Alzheimer's presentation. And so this issue is something that we consider as a multidimensional concept. One of the main factors we're interested in is the clinical syndrome, because we know that not all patients present with memory predominant deficits and some other phenotypes are associated with underlying Alzheimer's pathology. We also know there's a lot of variability in how old patients are when they develop their first symptoms, with some patients developing their first deficits in their 50s or 60s, even within sporadic AD. We know there's a lot of variability in how fast patients decline over time, and our research is really looking into the association between these factors in demographic, genetic, or neuropathological variables. So for the sake of this 5-minute presentation, I'll be talking mainly about the age of onset variability and how it relates to measures derived from neuroimaging or neuropathological studies. And I'll be presenting about 2 samples from the UCSF ADRC all including patients with MCI or dementia, were biomarker or pathologically confirmed for Alzheimer's disease. Here we're looking at the amount of amyloid or tau pathology in the overall cortex and how it relates to patient's age. We can see that unlike amyloid, tau is strongly related with age of onset, with younger patients having more severe tau burden. We saw that this relationship was not driven by any specific phenotype, as this relationship here can be found even in atypical AD, like so the visual spatial or language predominant forms. And we saw that this relationship was found mostly in the frontal and parietal areas, but not so much in the frontal lobe. So we're interested in seeing if this was specific to how we measured tau with PET, or if it could relate to a different type of measure. So we went to the brain bank dataset and used this quantitative measure of tau tangle density developed by Lea Greenberg and Katrin Petersen here at the MACC. And sampling 4 different brain regions, we could still see this very strong negative relationship with younger patients having more tau in their cortex. And like we did with PET, we did not find this association in the medial temporal lobe here in the CA1 and subiculum subfield. We're interested in seeing how this higher tau burden in younger patients was associated with disease progression. And so we used a subsample of the imaging study patients who had longitudinal MRI following the tau scan. We saw that this rate of brain atrophy over time was strongly related to both baseline tau and age of onset. And in a mediation analysis, we showed that younger patients had more severe brain atrophy over time, mainly because they had higher tau burden at baseline. Lastly, we looked in the brain bank datasets at the coexisting neuropathologies, because we know that Alzheimer's pathology rarely happens in isolation. And what we could see dividing the sample into early onset of late onset, based on the age of onset of 65 years old, we could see that a lot of pathologies were way more frequent in older onset patients. However, this pattern was not seen in for all neuropathological diagnosis, and patients with early onset have very frequent CAA and Lewy body disease. You can see even as frequent as the late onset. So to summarize, early onset Alzheimer's disease is associated with a greater tau burden in the cortex, but not in the medial temporal lobe. And the results were very similar, whether we quantified tau pathology with imaging in living patients or postmortem. We saw that this greater tau burden was associated with a more aggressive brain atrophy over time. But we also saw that early onset AD was associated with less coexistent neuropathological diagnosis. However, I want to insist on the fact that early onset is not either very pure disease, as CAA, Lewy body, and vascular brain injury were still very common. We think that these results are important because of how they can impact the development of clinical trials, both in terms of who we enroll in these trials and also what is the expected benefit from anti-amyloid or anti-tau treatment in different groups of patients. I want to thank you for your attention and thank everyone who participate to these studies, including our patients and their caregivers. Thank you.
Hello, my name is Shirley Wu, and I'm a 4th-year medical student at NYU Grossman School of Medicine. I'm really grateful to have this opportunity to present on my research on exploration of rapid automatized naming and existing visual tests for detection of early Alzheimer's disease. Alzheimer's disease, or AD, is often diagnosed in the dementia stage because it's at this point where symptoms become more apparent and disruptive. Biomarkers of AD precede clinical symptoms by many years, though, making it possible for detection at earlier stages, such as mild cognitive impairment, or MCI. In the past few decades, visual AD markers have been studied and have the potential to be sensitive and non-invasive. Rapid automatized naming, or RAM tests, have the potential to be sensitive visual measures because performance requires recruitment of a variety of brain pathways involved in different aspects of vision and cognition. Over here on the right is a Kindevic test, which is a rapid number naming test that has been shown to reliably differentiate MCI and mild AD from controls. However, as a whole, these types of tests have yet to be thoroughly explored in AD. Thus, our study objectives are to evaluate new RAND tests and measures of visual pathway structure and function for distinguishing MCI from cognitively normal individuals and see how these tests relate to vision-specific quality of life. So we performed a cross-sectional case control study where we recruited 15 MCI participants and 20 controls. We had them perform a variety of testing, including high and low contrast letter acuity, using the chart shown here, the Mobile Universal Lexicon Evaluation System, or MULES, a rapid picture naming test composed of colored photos of objects, fruits, and animals in context. The Staggered Uneven Number, or SUN test, which is a rapid number naming test shown here, composed of numbers arranged in unique patterns and with unique spacing. And participants are asked to quickly and accurately identify either the picture or the number that they see. This test is timed, and they are asked to complete 2 trials, and the best trial time and number of errors are recorded. Participants also underwent optical coherence tomography retinal scans, looking at thicknesses of their optic nerve head and macula, and also completed vision-specific quality of life questionnaires. So here we see that there are no significant differences between groups except for mini mental status exam scores, which are lower in the MCI group. Among all the different visual tests that we looked at, only low contrast letter acuity at 1.25% contrast, as well as MULES performance was significantly worse in the MCI group. You can see here that MCI participants were only able to see 40% of the low contrast letters that controls were able to see. And also, MCI participants took 1.6 times longer and committed 3 times more errors on the MULES test compared to controls. Worse 1.25% low contrast letter acuity, worse MULES times, and more MULES errors were significant predictors of MCI versus control status while controlling for ocular pathology. Interestingly, hippocampal atrophy on MRI was not a significant predictor of MCI when controlling for age. On ROC curve analyses, we show that these visual measures have good diagnostic accuracy of MCI versus control, with best MULES time having the best diagnostic accuracy. Furthermore, worse low contrast letter acuity at 1.25%, as well as more MULES errors were associated with worse quality of life. In conclusion, in this pilot study, rapid picture naming and low contrast letter acuity are sensitive measures that distinguish MCI from those with normal aging. These quick, easily administered tests are promising for identifying patients at risk for AD dementia and serving as measures in disease-modifying therapy clinical trials. In the future, we hope to assess these tests in larger cohorts and incorporate other types of retinal imaging. I would like to end by thanking my mentors, Dr. Laura Balzer and Dr. Arjun Masukar, as well as everyone at the NYU Neuro-Ophthalmology Vision Lab, NYU Alzheimer's Disease Research Center and Pearl Barlow Centers, as well as our collaborators in the NYU Ophthalmology Department and our current support. Thank you.
Here we examine how age-related changes in central nervous system inflammation, tau pathology, and synaptic degeneration impact sleep spindle expression and sleep-dependent memory in adults at risk for Alzheimer's disease. Fast frequency sleep spindles during non-REM sleep facilitate offline consolidation of memories by phase locking with cortical slow waves and hippocampal ripples to support neuroplasticity. Fast frequency sleep spindle activity is reduced over frontal cortex in older adults, and further reductions are observed in parietal derivations in MCI and AD patients. However, the mechanisms of these sleep spindle deficits remain unclear. One possibility is that core mechanisms of AD pathophysiology may underlie these deficits. The aim of the current study was to determine if age effects on fast frequency sleep spindles are associated with changes in CNS inflammation markers and their impact on AD pathology and neuronal integrity. Furthermore, we sought to examine if these deficits in sleep spindles were associated with impairments in overnight memory retention. 58 clinically normal older adults were recruited from the RAP cohort at University of Wisconsin-Madison. Participants were enriched for parental history of AD, but largely lacked amyloid and tau positivity. CSF was collected after an overnight fast, and markers of astrocyte and microglia activation, AD pathology, and synaptic and axonal degeneration were assayed. Of note, in healthy older adults, alpha-synuclein levels likely measure presynaptic degeneration instead of deposition within Lewy bodies. Participants underwent polysomnography with high-density EEG, and spectral analysis was implemented to examine fast sigma activity. Parallel serial mediation analysis was utilized to assess indirect effects. A schematic model is shown below and indicates that age X predicts fast sigma activity Y through its association with inflammation measures M1 through 4, and then AD pathology or neuronal integrity markers 5. Lastly, word pairs memory task was implemented with learning and immediate testing occurring prior to sleep and delay testing occurring after sleep. Topographic plots are shown on this slide. In all cases, warmer colors represent more positive associations, while cooler colors represent more negative associations. Light blue dots indicate EEG derivations with a significant association, while white asterisks show significant associations following TFCE correction. Green asterisks depict derivations with overlapping TFCE corrected significance for 9 CSF measures. First, all CNS inflammation markers were significantly associated with fast sigma activity, particularly over frontal EEG derivations. Next, associations were detected globally for tau, neurogranin, alpha-synuclein, and NFL. Of note, associations with alpha-synuclein were positive because a reciprocal transformation was applied. A conjunction of significant effects across 9 CSF measures were detected over 11 frontal EEG derivations, highlighted in green. Finally, associations were detected between fast sigma activity and the proportion of change in overnight memory retention, with associations at 11 EEG derivations overlapping with associations with all 9 CSF measures. We lastly examine the interrelations among all these measures to determine if age-related changes in CNS inflammation impacted sleep spindle expression through their effects on AD pathology and neuronal integrity. Age predicted soluble TREM2 levels, which in turn was associated with phosphorylated tau and alpha-synuclein. Each of these in turn was associated with fast sigma activity, and both showed significant indirect effects. Similar independent findings were detected through YKL-40, but not through S100B and GFAP. Trends were detected with TREM2 and YKL-40 through their effects on total tau and neurogranin. In conclusion, frontal fast sigma activity was most strongly associated with microglia-related markers, phosphorylated tau measures, and markers of synaptic degeneration. These spindle deficits were associated with impaired overnight memory retention. And importantly, these effects were detected in participants who were enriched for parental history of AD, but were largely amyloid and tau negative. Therefore, AD pathophysiology contributes to age-related sleep and memory deficits, even prior to A-beta and tau positivity. Future work should explore whether AD biomarkers impact longitudinal declines in sleep and sleep-dependent memory, and whether enhancing sleep spindles could limit memory decline in those at risk for AD. It is also important to determine if these associations change based on biomarker status, presence of sleep disorders, or are directly related to family history and risk factors. Lastly, it may be possible to arrest sleep-dependent memory decline in older age by targeting CNS inflammation, which may be an initiating factor. This study was made possible by the PIs Drs. Menkins-Benlin and their collaboration with Dr. Zenneberg and Mao. I want to especially acknowledge Dr. Sprecher, who led data collection as part of her dissertation. Finally, this work would not have been possible without the support from the National Institute of Aging.
Hello, I'm Valentina Ghisays from Banner Alzheimer's Institute in Phoenix, Arizona. Today, I would like to describe plasma beta 42/40 ratios in cognitively unimpaired, late middle age, and older adult APOE4 homozygotes, heterozygotes, and non-carriers from the Arizona APOE4 gene dose cohort. As many of you know, each additional copy of the APOE4 allele in a person's genotype is associated with a higher risk of Alzheimer's disease and a younger average age at dementia onset. My colleagues and I previously characterized relationships between amyloid and tau PET and MRI measurements and age in unimpaired participants with 2, 1, or no copies of the APOE4 allele from the Arizona APOE4 gene dose and the Mayo Clinic study of aging cohorts. More recently, we characterized plasma beta 42/40 ratios in 121 47 to 86-year-olds from the Arizona APOE cohort on their ability to discriminate individuals with an amyloid positive or amyloid negative PET scan. In this study, we wanted to characterize and compare plasma I-beta 42-40 ratios in cognitively unimpaired APOE4 homozygotes, heterozygotes, and non-carriers, including in those who remained unimpaired at older ages despite their genetic risk. We had 121 47 to 85-year-olds from the Arizona APOE cohort. Plasma I-beta 42-40 ratios were characterized using C2N's tandem mass spectrometry assay, and the ratio cutoff value is the actual optimal ratio retrospectively characterized. Age-matched subgroups included 80 47 to 70-year-old and 41 71 to 85-year-old homozygote, heterozygote, and non-carriers. We looked at linear associations with APOE4 gene dose in the overall and in the age-matched subgroups, and at the proportion of A-beta positivity across gene dose group. Here are the subject demographic characteristics. We had 21 homozygotes, 37 heterozygotes, and 63 non-carriers with similar educational level and sex distribution across APOE4 gene dose groups. Homozygotes were significantly younger than heterozygotes and non-carriers. We compared the proportion of amyloid positivity among gene dose groups in plasma I-beta 42-40 concentrations. As you can see, homozygote and heterozygote groups have a higher proportion of A-beta positivity and lower plasma A-beta 42-40 concentrations compared with non-carriers. Next, we compared the proportion of amyloid positivity among gene dose groups in plasma A-beta 42-40 concentrations in age-matched 47 to 70 and 71 to 85-year-old subgroups. As you can see, homo and heterozygote groups have a higher proportion of A-beta positivity and lower plasma A-beta 42-40 concentrations compared with non-carriers before age 70 but not after. On the right, we see that 3 out of the 4 cognitively unimpaired homozygotes over 70 years of age had normal A-beta 42-40 ratios, consistent with our previous findings using amyloid PET scans and suggesting that there are factors that account for resistance to A-beta plaque deposition itself in those APOE4 homozygotes who remain cognitively unimpaired at older ages despite their genetic risk. You can also see that APOE4 heterozygotes continue to have greater amyloid deposition at older ages than non-carriers, suggesting that a significant proportion of these individuals remain at higher risk of AD at older ages. To summarize, our previous amyloid PET findings and new plasma I-beta 42-40 findings demonstrate that APOE4 gene dose is associated with greater amyloid plaque deposition at older ages, and that plasma I-beta 42-40 ratios were inversely related to A-beta plaque burden and to the percentage of cognitively unimpaired homozygotes, heterozygotes, and non-carriers with a positive A-beta blood test in the overall and the younger age groups. It suggests that a resistance to amyloid plaque deposition may account for those APOE4 homozygotes who remain unimpaired at older ages. Additional studies are needed to clarify the potentially targetable protective factors that account for the absence of amyloid plaque deposition and cognitive impairment in those homozygotes who remain unimpaired at older ages despite their genetic risk. I would like to end by thanking the teams from the different sites and the funding institutions that made this work possible.
Hi, my name is Dan Ohm, and I'm a current postdoc at the University of Pennsylvania. Today, I'll be talking to you about the work I conducted during my PhD in the Meslem Center at Northwestern University. By way of background, I want to first introduce to everybody the clinical phenotype known as dementia of the Alzheimer's type, or DAT. DAT is the amnestic phenotype associated with AD and has a global atrophy pattern shown here in this MRI reconstruction. The vast majority of DAT patients have underlying Alzheimer's disease neuropathology, which is comprised of neurofibrillary tangles and amyloid beta plaques. AD is associated with both neuroinflammation and neurodegeneration, specifically an increase in activation of microglia and severe neuronal loss. AD is now associated with not just the amnestic phenotype, but also a whole range of other clinical phenotypes, one of which is known as primary progressive aphasia. PPA is considered a phasic variant of AD and has a unique signature pattern of cortical atrophy on its own, as shown here. What we see is more cortical atrophy in the left language network. Now, an interesting study by our group showed that the neurofibrillary tangles show an asymmetric distribution in PPA patients that's not seen in the DAT patients. In contrast, we see a rather similar distribution of plaques both in PPA patients and DAT patients. However, it's not well understood if patterns of neurodegeneration and inflammation have similar patterns to tangles in PPA patients. Furthermore, it's not well understood if these postmortem markers have a similar or have any relationship with cortical atrophy patterns in PPA. Establishing these relationships is important though, because MRI is becoming increasingly used as a reliable biomarker to identify and differentiate these different clinical phenotypes. Therefore, the current study set out to determine the biological correlates of cortical atrophy in PPA patients with AD. We had MRI scans close to death within 2.5 years, and we measured cortical atrophy and postmortem markers using stereology in the same regions of interest, which included language regions shown here in shades of blue, and in non-language regions shown here in shades of green. We established correspondence between these MRI-based regions and postmortem regions, as shown here in the schematic. Basically, we delineated regions shown here in white in an MRI coronal section and found matching postmortem coronal sections one at a time for all available stained tissue, and delineated those regions one section at a time. As to be expected for PPA, we see cortical atrophy lateralized to the left hemisphere and specific to certain regions within the language network. Similar to this cortical atrophy pattern, we see that neurofibrillary tangles also show that leftward asymmetry and regional selectivity to language areas that we did not see for plaques. Importantly, we found that there was a significant relationship between increasing severity of atrophy and the increasing densities of neurofibrillary tangles that we did not see with plaques. Together, this suggests that cortical atrophy and tangles accumulate in a way that has left language region selectivity in PPA patients. Furthermore, it suggests that tangle spread and propagation may deviate from conventional Brock staging, that limbic to neocortical staging, such that the PPA patients have a different distribution or propagation. Next, in our next set of experiments, we sought to determine the relation between AD pathology and markers of inflammation in neurons. First, we saw that the increasing densities of tangles were associated with increasing densities of activated microglia. We did not see a relation between plaques and activated microglia, however. Next, we determined that there were smaller neuron densities associated with not just increasing tangles, but also increasing activated hypertrophic microglia. In conclusion, we find that in with AD neuropathology, that the tangle densities, not plaque densities, mirror the cortical atrophy consistent with the aphasic profile of PPA. For neuroinflammation, it was activated microglia that may be more responsive to the tangles than plaques. And we did see we saw severe neurodegeneration in PPA patients that were associated with increased densities of tangles and activated microglia. Together, this gives us converging evidence that tangles and microglia-mediated inflammation may be important determinants of the degenerative processes that lead to the atrophy patterns, not only in PPA, but maybe the greater clinical spectrum of AD as well. And with that, I want to acknowledge the entire supportive staff at the Meslom Center, and in particular, my primary mentors, Emily Rokowski and Changiz Gurula. Thank you for your time.
Hello, my name is Robert W. Turner II, and I'm presenting today on behalf of the Boston University ADRC. And the title of my presentation is Conducting Community-Based Research with Black Men in the DMV to Examine Dementia Caregiver Burden. A growing body of literature suggests Black Americans have a greater likelihood of experiencing Alzheimer's disease compared to non-Hispanic white population. The number of older Black Americans with ADRD who will require some type of care is projected to triple in the upcoming decade. Now, although Black Americans are at greater risk of caregiver burden, the body of research that focuses on adult Black American men as primary caregivers of a person with dementia is sparse. And for our study purposes, the definition of a primary caregiver is someone who provides 10 hours or more of unpaid care for a person, a loved one. Our study is guided by the following question. What is the effect of dementia caregiving stress on cognitive function in adult Black American males? The two aims of our project are: one, examine which aspect of cognitive performance are affected by caregiver status and stress. Aim two investigates which stress-related physiological and self-reported psychological measures may mediate the caregiver effect on cognitive performance. The goal of our project is to recruit 40 caregivers and a comparison group of 40 non-caregivers, which include Black American males ages 30 to 85 years old who speak, write, and comprehend in English with a CESD score of less than 16, living in the DMV area, which consists of Maryland and Virginia, areas that are close to Washington, D.C. Our initial pre-COVID recruitment strategies included in-person recruitment at community organizations and in-person recruitment at local DMV community events, as well as the use of social media. Our current strategy post-COVID includes emails and calls to universities, faith-based organizations, and community organizations in the local DMV area, along with virtual presentations to those organizations of our study, with an extensive use of social media as well. Our methodology includes concurrent community-based mixed methods research approach, and data collection methods include health questionnaires and the uniform data surveys, along with a neurocognitive battery of tests. We also ask participants to participate in in-depth recorded interviews where they will be asked questions about their experiences with caregiving and about their roles and relationships and their routine, and even if they have any recreational or physical activities. We also collect data by asking participants to, they receive saliva kits along with instructions on how to use those over the course of 2 days at 2 different time periods. Now, of course, with any project, there are some challenges. And pre-COVID, our challenge was to collect questionnaires and assessments in person. Now, post-COVID was about how do we now collect those saliva samples? And one of the things that we learned during our interviews is that Black men are very concerned about providing saliva samples because they want to know who will have access to their DNA, will police or other government agencies. So we've had to reassure them that because of our safety protocols, no one will have access to that. Now, because of COVID, we've had to modify our data collection procedures. Post-COVID, we now mail our saliva kits to individuals. We also have physical distance and study participant safety plans in place, which involve where we now drive to participants' location and we collect those saliva samples are placed in portable coolers outside of a person's front door. We have no person-to-person contact, and we also produce a instructional and informational videos on how to actually utilize the saliva sample kits appropriately. With that, I'd like to thank you very much for providing the opportunity to present at the Spring 2021 ADRC. And as you can see on the screen, my email is provided to you. And again, thank you, and please reach out with any questions.
My name is Karina Alviña, and I'm a research assistant professor in the Department of Neuroscience at the University of Florida. My talk today is about irisine as a mediator of exercise-related cognitive improvement of hippocampal function. Exercise is good for your brain, as we probably know. Some of the effects of exercise that are depicted here are very important. One of them is an increase in neurogenesis in the dentate gyrus of the hippocampus, which is one of the regions in the brain where there is newborn cells born in adulthood. And together, there are other structural changes, such as increasing the dendritic spines and plasticity, all in response to exercise. Another effect that is very important is the increase in neurotrophins, in particular BDNF, or brain-derived neurotrophic factor, which is really important for growth and development of the brain. And finally, another important effect that we are going to revisit now is a reduction in the A-beta burden in the hippocampus. So irisine, or exercise could have important effects for neurodegenerative disorders. So irisine comes from the skeletal muscle depicted here in this cartoon, and from a precursor called FNDC5. Upon exercise, that protein is cleaved, and irisine, which is a segment of the protein here, is released to circulation. The first effect that was described was actually in the context of metabolism. Irisine drives browning of adipose tissue. So this is white adipose tissue, and when irisine binds to a receptor that was recently identified, that adipose tissue browns and increases energy expenditure and thermogenesis. A couple of years later, it was also shown that FNDC5 and irisine can actually have effects in the brain. And this is an important factor that we're going to take into account. If mice are subjected to a running protocol, like shown here, FNDC5 increases in the muscle as expected, but also in the hippocampus of these mice. And the hippocampus is the area of the brain that is related to memory and learning. And importantly, BDNF, as shown here, also increases in the hippocampus of these mice. And BDNF is good for growth and development. In fast forward a couple of years, in this very important paper came out that showed that FNDC5 and irisine are actually reduced in Alzheimer's disease samples. So what we have here is the Western blot for FNDC5 and irisine in controls, early Alzheimer's disease patients in late Alzheimer's disease samples. And as it is quantified here, the late samples show a significant decrease in the expression of this protein, which was confirmed later with samples from CSF from patients. This is healthy controls in mild cognitive impairment patients, which did not have any significant difference, but Alzheimer's disease and Lewy body dementia patients showed a significant decrease, which was not observed in the plasma. So this was exclusive for CSF. That led us to think that irisine could have an important effect as neuroprotective. And we're going to focus on cellular and synaptic function, specifically targeting hippocampal cells in an animal model. And this is going to be important later once we get our colony going here to test this hypothesis specifically in an Alzheimer's mouse model. To detect changes caused by irisine in the brain, we're going to use electrophysiology. These are acute slices from from brains of mice. We section them and we put them in a microscope with electrodes to record the electrical activity in the hippocampus. Our preliminary data shown here, these are field recordings in control, and in the presence of irisine for 1 hour or 3 hours, shows that the longest the incubation with irisine, the larger the field potentials. Even though these are preliminary, we really think that the treatment with irisine increase the size of this field potential, so it could have protective effects. We're currently working in understanding what mediates this effect. And of course, the final idea is to look at this in the context of an Alzheimer's model. And if you want to know more about irisine in neuroprotection, we recently published this very comprehensive review article showing all the possible ways in which irisine can be neuroprotective. And with that, thank you.
Right, I just want to thank all of today's speakers and as well as everybody who recorded their presentations. Please make sure to check out those presentations. The link is in the chat, and make sure to vote for the top speakers, which will be really difficult because they were all amazing. But please go ahead and make your votes. And make sure to join us tomorrow morning to see some more of these wonderful presentations. Thanks, everyone.